So hi and hello everyone. So today we are from group 6, BSM 3201. We are going to do a group discussion. So before we start, let me introduce myself first. My name is FAIZAH binti ZAINAL ABIDIN and my matrix number is 208074.
My name is AMIRAH FARZANA binti ISMAIL and my matrix number is 208194.
My name is FATIHAH binti AB.RAZAK. My matrix number is 205360.
My name is NURAINI binti MOKHTAR with matrix number 205304.
My name is FARAHAH binti ZUHARI. My matric number is 208094.
So today we are going to do a group discussion for the replication of BNT. We need to illustrate the replication of the following DNA strand end to end. So DNA replication is the process by which the double-stranded DNA molecule is copied to produce two identical DNA molecules. So Amira, would you like to explain first about the DNA replication, about how it started?
Sure. But first, Faizah, can you zoom in a little bit?
Okay.
Okay, thank you. So I think the replication will start with initiation first. So according to the question, as we can see, there is two original replication. So from this, we knew that this is a eukaryote, okay? Eukaryote chromosome lah. Eukaryote chromosome. So in the initiation, there is the formation of replication fork. So what is replication fork? Replication fork is the junction where there is replicated DNA and the unreplicated DNA. As you can see, there is a picture under the question. So this is the replication fork part. There is replicated DNA, then there is unreplicated DNA. So, okay, the origin, we start the initiation at the origin of replication. So there is two ORIC there. So the initiation, we start at the both part. And then the replication will be bidirectional replication, which means that they will move like this. As you can see my hand, like this. Okay. So when they are moving like this, both part will move like this. So they will meet at the center. So they will fuse together, and that's why, as you can see, there is a fuse replication like that, a long replication. So I think I only know that part. Is there anyone to continue? Maybe she can explain about the protein in the in the initiation.
Okay, alright. So the overall idea for initiation is the assembly of proteins and enzymes in replication fork, right? So maybe in synthesis always initiate at origin of replication uses the specific loci in the DNA genome, which high concentration of AMP vector. So there are specific chromosomal locations called origin of replication where replication begins. So in eukaryote, specifically, origin of replication are defined as nucleotide sequences that bind to the origin recognition complex, which is ORIC, which initiates replication. And then eukaryotes have linear chromosomes and they initiate replication at multiple origins. But for prokaryotes, on the other hand, have much smaller genomes which are circular and they have only one origin of replication. So it starts with DNA helicase, which unwinds DNA hydrogen bonds between two strands of DNA. So the unwinding resulted in replication forks that flow in both directions, away from origin, forming replication bubbles. So DNA synthesis proceeds in 5 to 3 strands. Okay, once separated, SSB, which is single-stranded binding protein, binds to single-stranded DNA strands. So binding of one SSB promotes another SSB, binds to adjacent SSDNA, which is single-stranded DNA, and interacts with the bumped SSB to prevent re-ending. Then the requirements of RNA primer to initiate DNA replication by DNA polymerase makes RNA polymerase synthesizes short RNA primers using single-stranded DNA template, and then the primer is being removed by DNA polymerase 1 and replaced with DNA. Then DNA ligase joins the two nucleotides between the two Okazaki fragments by forming a phosphodiester bond. Yes, that's it
So
regarding.
Farahah,
Yes,
so DNA synthesis will uh, form the strand from where to where?
5 to 3 strands.
So how about the if both strand have to synthesis simultaneously? How about the 5 to 3 template? Is it also produced the same way as the 3 to 5 template?
Okay. All right. If both strands were to be synthesized continuously while the replication fork is moving, how to synthesize? The 5 to strand one strand will have to undergo 3 to 5 synthesis, and it will be synthesized in lagging strand, forming the Okazaki fragment.
Uh,
Okay.
Since my friend already discussed about initiation step, now I will explain about elongation, especially about the catalytic requirement. So elongation start when the first phosphodiester bond formed between RNA primer and the first nucleotide base, and this process is dominated by DNA synthesis. So there are two catalytic requirements for this process uh, that's supposed to present. The first one, which is deoxynucleoside triphosphate. It is the ATP, the TTP, the GCPP, and the CPP. So basically, the deoxynucleoside triphosphate consists of deoxyribose sugar, nitrogenous bases. It's either adenine, guanine, cytosine, and thymine. As we know, this is for DNA synthesis, and three phosphate group that are bonded together. Now, its function as monomer sDNA, as well source of energy to initiate and sustain DNA synthesis. The second catalytic requirement is a primer or a template junction. So the OH group at the 3' end of the primer is important in DNA synthesis because the 3OH will be extended by nucleotide addition. So DNA synthesis is initiated when 3' of hydroxyl group of the primer mediates the nucleophilic attacks at the alpha phosphate of the incoming DNTP. So this results in extension of the 3' primer by one nucleotide and release one molecule of one pyrophosphate. So the hydrolysis of this pyrophosphate will uh, result in two release of two phosphate molecule. And next, FAIZAH will explain in detail how DNA is replicated.
So thank you, Fatihah. So basically, Fatihah just said that DNA is synthesized by extending the 3' of the primer by DNA polymerase. So DNA polymerase, it will wrap itself around the strand, and then it will attach the new nucleotides to the nitrogenous bases that are exposed, which is the single-stranded DNA. So in this way, the polymerase will assemble a new DNA strand on the top of the old one, like this. So this is the new one, and this is the old one. So as DNA polymerase move down the unwound DNA strand, it will build the new strand from the pool of free protein nucleotides, which is the NTP, like FATIHAH just explained just now, that surround the old strand. So the new strands nucleotide are matched with the partner nucleotide in the template strand, where the adenine will pair with the thymine and cytosine will pair with the guanine. And this phenomenon is known as the complementary base pairing and result in the production of two complementary of DNA strand, like this. Um, as the result of the complementary base pairing, replication continues as a succession of sequence and anti-sequence copies, preserving the original DNA code. In other words, DNA replication is semi-conservative where it conserves the old strand. And on the leading strand, as you can see here, it has a leading strand and the lagging strand. Yeah. So the leading strand proceed continuously, but on the lagging strand, it proceed discontinuously. And we can see that the break, or we call it nick, to produce the leading strand like the this one. So DNA ligase will bind and help to seal the break by forming phosphodiester bonds. And yeah, replication and
I'm sorry to interrupt FAIZAH, but before the replication end, you need to undergo the termination step.
oh, could you explain more?
Okay, basically in this termination step, at some point at the lagging strand, the primase cannot synthesize the RNA primer more because of sufficient of space. So it will causing the incomplete replication and the forming of the short ssDNA at the region at 3N. So how to solve this problem? We have the telomeres. So at the end of the linear chromosome, there are spatial region we call as telomeres region, where it contains the tandem DNA repeat, and it is a region where the replication of DNA will occur, which it will catalyzed by the telomerase. Can you scroll down there?
Sure.
Okay, thank you. So basically, the DNA replication at the telomeres region, as you can see from the drawing, it has 3 steps. Firstly, the binding, where the telomerase it will bind to the 3' overhang at the telomerase region, and then the telomerase it will synthesize the 6 nucleotide repeat. And then moving on into the second step, the polymerization, where the telomerase it will move 6 nucleotide to the right and begins to make another repeat. And lastly will be the translocation step, where the complementary strand is made by primase, DNA polymerase, and ligase. And then in the end, it will form a complete gene sequence. I think that's all for me.
So basically, the telomerase will facilitate in the termination, right?
Yes.
Okay. So in conclusion, we can say that DNA replication occur in 3 stages, which is the initiation, elongation, and termination. Basically, the replication will form at the origin, just like the AMIRAH said. So it will form the replication fork here, and the primer will be synthesized to initiate the RNA synthesis, DNA synthesis. Then the DNA polymerase will synthesize the DNA from the primer in 5 to 3 direction. And finally, the telomerase will facilitate in termination and produce the complementary complete complementary strand. So I think that's all for our discussion. I think we have reached the end of our discussion, and thank you for participating, and have a nice day, everyone.
Thank you, everyone.
Thank you, everyone.
Thank you guys.